So I'm Carlo Maria Carbonaro. I'm from the Department of Physics of University of Cagliari. Thanks for this opportunity to all the organizers, in Sergei in particular. I'm going to talk about carbon dots and the way we combine experimental and computational approach in order to have some insight in their structure and properties. I have to say that carbon dots are a sort of hybrid system where we have both molecules, crystals, and order and disorder systems. So they are quite funny to simulate and to try to understand how they work from the experimental point of view. I'm too an hybrid people because I am both performing both the experiment and the computation, not only by myself, also with people working in my group. So my presentation is the following. I start with a short presentation of carbon dots in the sense of what they are and how we prepare them and which are their properties, why they are so interesting for the possible application in technology, and which are the open issues that we still have to solve. Then I will move to the photophysics, since we need to understand what photophysics is for carbon dots. I will just give a brief presentation about basic concepts and application, and which are the experimental setups that we use to characterize, in particular, carbon dots, but they can be used for everything indeed. I will briefly discuss the computational modeling, that one which we use in order to understand the carbon dots, but of course, the previous talks in these days have been more complete than my presentation about these possibilities. However, I will describe some complex systems and some application to CDs. Then I will move to the synthesis and characterization of carbon dots, looking at the experiment that we have done, not only optical, but also NMR and others, and then to the density functional simulation and molecular dynamics simulation that we have performed in order to understand some things about our samples. So let's start by talking about carbon dots. Carbon dots are basically zero-dimensional materials. They have more or less 10 nanometers the main size is 10 nanometers in diameter. They are usually represented, this is just a picture, as possibly as graphene layers with a lot of dopants and atomic groups on the edge, but also inside the layers, but also different. This picture here is another one. I mean, it's just a model because usually we have also a lot of paddle of disordered carbon network. They have a very interesting photoluminescence in the sense that the luminescence can be tuned by changing the excitation. So we can move from the blue till the red and the infrared emission with different, of course, quantum yield, which is quite high in the blue and the green, a little bit less in the region of the red, but we are working on that, of course. So we can have different kinds of carbon dots with high quantum yield of emission. We can also prepare them from organic molecules or polymers, and also from renewable sources. So they are in principle green, non-toxic, and cheap. So it means that they can be very interesting in order to substitute the quantum dots, which are now reused for optical applications and for imaging application, for example, in bioimaging systems, but they are quite toxic. So we would like to substitute carbon dots, quantum dots with carbon dots, and then to apply the optical future in different high-tech fields like bioimaging, like sensing, optoelectronics, producing light, or environmental fields. So for the sensing, for example, of by changing the pH, or to sense the presence of methylene blue, to treat the water in order to remove the pollutants, and so on. Because carbon dots are also very dispersible in water, so they can be applied in biological systems. Well, how do we prepare our samples? Well, we can use two different approaches. One is called top-down approach, and the other one is called bottom-up. They are quite different also in the results they give. The top-down approach is obtained by taking some carbon resources like, for example, bulk systems, and then we cut them, for example, by laser ablation, electrochemical treatment, or arc discharge, and then we get the carbon dots. In that case, usually they are quite ordered because the starting system is an ordered one. Another approach, the one that we usually use because it's more easy and also can be performed with less impacting experimental stuff, is the bottom-up approach. When we take some organic molecules, we use something to make the pyrolysis, for example, hydrothermal treatment, ultrasonic, or microwave. And then we get our carbon dots, which are at the end a bit more disordered than the other ones. So there is also in the academic and in the academy and in the literature, there is still the need to define different kinds of carbon dots, because under carbon dots is a big umbrella where we have a lot of different nanoparticles, which can be different because of the carbonization degree and also because of the graphitization of our system. So we talk about carbon quantum dots when there is a quite high degree of graphitization, and on the other side we distinguish between carbon nanodots, carbon polymer dots, and polymer dots when we change the sp2 sp3 relative ratio in order to get some order and disorder system in the same nanoparticle. So starting from biomass, starting from polymers, we can assemble our system in order to have a carbon nanostructure, which can be quite difficult to represent. And this is one of the issues that we want to solve by computational simulations, of course, and also by experimental approach when we look at these nanoparticles by, for example, TEM and so on. And then we move to understand why this structure is so related to the optical feature, how it is related. Because, of course, depending on the structure, we have also different optical features. For example, in the case of very ordered system, usually we apply as a model for the optical feature, we observe the quantum confinement. In other cases, when we have a system with a lot of different edge functionalization on the surface, like different edge chemical groups, we call for the presence of surface states, which modify the electronic level systems inside our nanostructure, giving origin to some surface states, which could provide both the absorption and the emission properties that we get from the experiment. Finally, since we start from some organic precursors in the bottom-up approach, in that case, it is quite easy to obtain molecular fluorophores. This is the case that we are interested in, since we start from usually citric acid and urea in order to produce our samples. So what we are interested in is in the presence of this kind of fluorophores and how we can change the kind of fluorophores by changing the precursors and their relative ratio. In particular, we are interested in the production of blue-emitting fluorophores and green-emitting fluorophores. In the follow, I will call them citrazinic acid-like fluorophores and HPPT-like fluorophores. And of course, what we have to understand is how these molecular fluorophores are bonded to the surface or are embedded within the carbon network of our system. So the idea is that the optical features that we get are related to these molecules. So we need to investigate these properties both from the experimental point of view and the computational point of view. Some basic concepts about the photophysics, which is the experimental part I usually perform. Very basic, I'm talking about the interaction of the light with matter, so the various way in which the light can interact with different materials, and all of you knows what is an electromagnetic radiation, and the way this electromagnetic radiation can interact with materials is through different mechanisms. We can have the absorption, we can have some excitation of luminescence, we can have a non-radiative transition, excitation formation, exciton formation, relaxation system, and so on. So photophysics is basically the study of how the light interacts with matter, both at the atomic and molecular levels, usually specifically because we are interested in the processes, physical processes that occur without chemical changes. So we are looking at absorption and emissions, and emission of light, absorption of light and emission of light by the material, and energy transfer in between some molecular groups or molecular fluorophore and so on, which are inside, or the effects in case of more complex systems, which are inside our material. Well, everybody knows that when we look at the atom or at a molecule, we can describe the electronic system by considering the formation of the ground states of the electronic states that can be occupied according to quantum mechanics rules by our electrons. And of course, when we put all the atoms together, we get a very complicated system which can be both ordered or disordered. In that case, we obtain the formation of bands, which are known as the conduction and the valence band in the case of the dielectric systems, where we have a separation in between the two bands. The separation decreases when we go from dielectrics to semiconductors, and of course, these two bands are completely overlapped in the case of metal systems. So we can study the formation of these bands, which is also quite complex depending on the space we are using to represent our bands, and this is how we try to understand the optical feature of our system. Some easy keywords that we use usually when we are talking about molecules, and we are talking about molecules inside carbon dots. We have, of course, the absorption spectrum, or the reflectance and the scattering spectra, which are related to this same concept, where we have the transition from the ground state of our molecules to the first or second and so on level of the molecule itself. Then from that, we can have the production of different light emitted by the molecules. For example, we can have the fluorescence after the relaxation from the excited state from the upper states of the S1 to the bottom level of this electronic system down to the ground state. We can have also the transition to the triplet states in order to get the phosphorescence, which is just a longer emission of light. Of course, we have to talk about luminescence lifetimes, which allow us to understand which is fluorescence, which is quite fast, and which is phosphorescence, which is quite which is not fast at all, which is in the millisecond regime usually. And then we have to talk about luminescence quantum field. That means how many photons we get after the absorption of the light. And of course, we can also try to understand how is the excited state dynamics, which means to look at the vibrational relaxation of all the excited states, or the way we go from the singlet to the triplet states, or from the S2 to the S1 states, in order to understand which is the scheme of the levels that we have to consider to explain the optical properties of our molecule. There are different factors which are influencing the photophysics properties. First of all, the structure of the material. Of course, if we are dealing with the electrics or semiconductors, metals, or if you are dealing with bulk materials or nanocomposites, we have, of course, different behavior of the interaction of light with matter. There are environmental effects that we have to take into account. For example, in the picture, you see a fluorophore, which is based on isocyanate phthalate, and the optical properties of this molecule change from the blue, deep blue, to the green by changing the solvent. So there is an environmental effect due to the presence of different solvents, or we can also have some aggregation-induced effect. Then we have possible intermolecular interactions, so we can have both the excitation and the charge transfer interaction, as here depicted for the fluorescein derivatives with electron donating different substituent group on the benzene moiety. So, and finally, of course, we could also consider the spin-orbit coupling in order to understand the phosphorescence mechanism and the energy transfer between different excited states. So these are the factors which influence the photophysics of our systems. The applications are quite high, so we can consider, for example, light-emitting diodes in sense of organic LEDs and LEDs, which are applied on our day life or nowadays. We can consider also the use of luminescence and materials and photophysics for sensors and biosensor. Here you see how we start from the sensor samples. We go to the transducer to produce an electronic system which is able to detect some biomolecules which are interested for our application. We can use our materials for the photocatalysis and the photochemistry in order to produce new energy sources, or to allow our system to remove pollutants from water and air. And finally, we can also consider the application of photophysics for bioimaging. In this case, for example, we are using up conversion in order to excite some system in the very far infrared because of the possibility of light to go deeper inside the skin of our mice, and then to excite some system in order to get the imaging of the tumor we are looking at or we want to deal with. Okay. Usually, the representation of fluorescence and phosphorescence is given by a configuration coordinates plot where when we represent the energy of our levels, and then we have the absorption, the relaxation, from non-radiative relaxation from the system down to the bottom of our parabola, which is of course an approximation of the band, and then we go back to the ground state by emitting some fluorescence in terms of fluorescence or phosphorescence, depending on the time that this emission takes. In particular, phosphorescence is has a quite longer time. There is a persistent of the glowing, so we have the emission, and all of us knows that we could have phosphorescence even for seconds or minutes after the excitation light is removed. On the other hand, if you will look at the fluorescence, well, we have the Stokes shift from the absorption to the fluorescence, and then the fluorescence is quite immediate. I mean, is a very fast process which happens in the order of the nanosecond, while the internal conversion and all the intersystem crossing usually, or the electron transfer, usually are in the femtosecond and picosecond scale. So we need some experimental approach in order to differentiate by time these different emission properties of our molecules, which usually are represented, and all these properties are usually represented in the Jabrowski diagram for fluorescence and phosphorescence that I'm sure you already know. Okay, so which are the experimental setup that we usually use? We can use some steady-state optical absorption spectroscopy by using a double beam system, or we can use an excitation emission spectroscopy system in order to get both the map of the excitation and the emission properties of our system. And these are steady states characterization. We also are able to perform some excited state dynamic by transient absorption spectroscopy. This is quite more complicated stuff because we need to have some femtosecond laser. We need to pump and probe our sample at different delay time. When I'm saying different delay time, I mean that the light from the pump and the probe are arriving on the sample in the same position, spatial position, but at different time, and the difference in time could be from few femtoseconds up to some nanoseconds, usually, at least for the samples we are interested in. Or we can also measure the decay time of the emissions, which means that we are making some time-resolved emission spectroscopy, usually by some gated CCD camera. So we are pinging on the sample by a laser, which is not a continuous one, but is a repeated. We have a repeated pulses of the laser, and then in between two repeated pulses, we are analyzing how evolves the emission from the system. We can get the information by using a window which is gated and this is moving across the time, or we can get the same information by using some street camera, where we have on the image on your left, you have both, on the map, you have both the wavelength and the time, which means that we are getting both the information at the same time. Again, by using a femtosecond on nanosecond laser, a pulsed femtosecond nanosecond laser, in order to have the excitation of our system, then a monochromator in order to get the wavelength, the dispersion, and finally the streak camera in order to have the time dispersion of the emitted light. Okay, so this is the basic of the experiment we perform, at least from the optical point of view. And then some few words about computational modeling. Dirac said years and years ago that if we could be that we know all the law necessary for mathematical theory, and the rule of chemistry are completely known. But the difficult now is to apply these laws to solve this equation, not in very easy, simple system, but in our very complicated daily life system, as is a carbon dot, of course. So the computational chemistry and physics concern with theoretically determining structures and electronic properties of materials. And since it's difficult to deal with nanosized materials, like, for example, our carbon dots, computational modeling in these years has become very, very important as a characterization tool for nanotechnology. Well, the starting point is the Schrödinger equation, and if the equation could be fully solved, all information which pertain to the system could be determined, which means that we have to solve this complex equation, but they are not solvable for systems with very many atoms. And since these are not solvable, we need the help of computer used in conjunction with some simplification. We have already seen some beautiful presentation yesterday about that, and the parametrization that we need to solve the equation. For examples, we can describe both the wave and the particle behavior of electrons. The wave function is described by the wave function by the psi, while the particle behavior is represented by the energy. And in system when we have more than one electron, the wave function is dependent on the position of the atoms, and we need to describe in an accurate way the geometry of our system. So we need to solve this equation. I will leave this link if you want to have some nice way to understand how we go from the classical equation of the energy till the Schrödinger equation. And once we have a model, once we have solved, then we have to construct the model in order to solve the equations and determine the energetics of our system, in particular our molecules, in the case I'm presenting, but of course this is for all the systems. So the different models use different approximations, which means different levels of theory, in order to obtain some results with different accuracy. So we have a sort of balance in between cost calculation and accuracy, and depending on the system that we are taking into account, we can go from the atomistic molecular mechanism till some correlated methods, as we have seen yesterday, and we have to make a trade-off between accuracy and computational time. There are basically two types of models: the one that use the Schrödinger equations or simplification of the equation, and thus that do not use this equation. So we can distinguish in between ab initio, semi-empirical, and molecular dynamics. I'm not going too much into the details of all of that. I just want to give you a brief idea of what they do, so we can understand, we can represent our system. In this case, I'm showing some picture of papers which have which were dealing with the representation of carbon dots. So constructing, building some system in order to get optical properties of carbon networks able to represent the optical features of experimental carbon dots. And in this case, they were using ab initio models. Of course, we can have some mixed system where we have a very accurate system, like an onion system, where we have a very accurate system where when we are interested in the optical feature of a small portion of our system, then we are losing some step of accuracy going to a larger shell, and then finally to a very larger one in order to represent all the possible interaction with the, with the surrounding, as in the picture shown on your right. Okay, so we have to choose which is the molecular models you want to apply, depending on what we are looking for the tip, for the future we want, or the structure we want to understand of our system. So we start in the case of ab initio from the very beginning by using some first principle approach. In that case, we do not use any experimental data. We can have different levels of ab initio calculations, like the R3 fog. We have seen yesterday the presentation about that. We can have some Moller-Plesset perturbation theory, density functional theory, which is the one that usually I use, and the conflection interaction, in order to get different models, different levels of accuracy also in these systems, depending on the complexity of the system, on the accuracy and the property we want to look at. We need to use some approximation as the central field approximation or the LCAO we have seen yesterday, so I skip it. And basically, we distinguish in between three basic types of calculation where we can get the information we are looking at. Single point energy calculation, which predict the stability and the reaction mechanisms. The geometry optimization, which predict the shape of our system, like in the case of carbonium 6T. Then the frequency, which allows us to predict the spectra, infrared spectra, Raman spectra, or in some case, the calculation of the absorption and the emission of our molecules. Considering an ab initio method, density functional theory is a way, as we have seen yesterday, to solve the problem from a different point of view, approaching the problem by considering the density of the systems. There are three density functional theory calculations. One is using local density approximation that we know is fast, but is very not accurate for the geometry. We can use the gradient corrected approximation, which gives more accurate geometry. So we can use, as we usually do, some hybrid approach where we have a combination DFT and R3 methods, which are able to give more accurate geometries and also quite good optical properties in the case of some molecules. The same empirical method uses experimental data to parameterize the equations. So we have a faster system, and we can increase the dimension of our systems. So they are faster but less accurate, of course. And they usually reproduce some specific results according to the parametrization and the parameters that we have using. In this case, for example, it was applied to model disordered carbon dots in order to get the idea the idea were the presence of some edge groups, which means some chemical groups at the surface of our carbon dots, gave some electronic levels inside the band gap of our system. And we have seen yesterday that we can use different basis sets in order to represent to have a mathematical function in order which is able to describe the shape of the orbitals of our systems, which could be quite easy in the case of H2 molecule, but of course it's very difficult in the case of more complicated systems and molecules, the one that we have. So usually we use some approximation even in this case where we use not only the Slater type orbitals, but also the Gaussian approximation and the Gaussian functions. Okay, I will skip all this data just to. Yesterday it was shown that all the names that we have are referred to the way we are approximating the description of our system, both from the basis sets and also in the choice of the hybrid pseudo potential. And finally, we can use also molecular dynamics in order to get some interesting and very nice description of our system, calculating all the velocities of the atoms, the forces involved, and then solving the system with a very large number of atoms inside, I mean thousands of atoms, in order to get the density plot, the interlayer spacing of our structure, and a molecular representation of our carbon nanodots. Again, we are choosing in between the length scale of what we are representing and the time scale of what we are representing in the terms of also computational cost. And finally, we can use also multi-scale simulations in order to have a combination of all the information to get some more accurate description at different level of our length scales. Okay, let's conclude by saying that we have the Schrödinger equation. We can solve it, but we cannot solve for very complicated systems. So we need to use some approximation of this equation, and the level of accuracy and computational time of the simulation depends on the model and the basis set that we are using to represent our system. So in the case of carbon dots, there are a lot of paper dealing with the structure and the features of carbon dots in these last 20 years at least. Usually the approach is not to predict but to explain what is going on, even though in these last years the approach is changing because we want to have the help of computational chemistry in order to predict the properties that our carbon dot should have for some specific application. So we can also in this case consider the optical features of carbon dots, how they are affected by the structural organization, that by the presence of some elemental doping, by the size and other factors which are fundamental to rationalize in order to get a design and technological application of the system. Here you see some very nice application you can find in the literature in order to understand the optical features of carbon dots, where you see that we have to consider in your system, for example, the contribution of sp2 and sp3 carbon in the carbon network. You also have to consider the planarity and the topology because of the presence of edge emissive centers, which changes the optical properties of your system. And you have also to consider the presence of some molecules which can be inside or outside your carbon network. Usually the carbon nanoparticles is represented as a core shell systems, which means that in principle we have a pure carbon network inside and a shell with a lot of edge groups and molecules outside, but it could also be possible to have some molecules inside the system, which is embedded within the nanostructure. So the idea is to understand how these different effects affect the optical properties of the carbon dot. The idea is to understand how the structure affects the optical of carbon dots, affects the optical properties of them. Indeed, we see that if we increase the size, we move from blue emission, or let's say we are usually talking about optical properties, so where we have the emission of our system. So we can have a blue shift of the optical properties by changing the planarity or increasing this P3 to SP2 ratio, or a red shift of the optical properties by increasing the size. And also we have to consider the presence of different molecules and doping elements, which allow to modify the electronic levels in the systems. But the still open question is how we can explain the formation of our system, because we know that if we start from the bottom-up approach, we start from some very basic organic molecules, we mix them, and then we heat them at some temperature, and if we increase the temperature or we increase the synthesis time, we change the ratio in between the molecular the molecules which are contributing to the optical feature, or the carbon core which is contributing to the optical feature of our system. It was really recently shown that it happens in the very few seconds of the synthesis. So we need to understand how we go from some molecules to the carbon structure, and how they are interacting with the structure within the system we are trying to understand. So the approach was to model the early stage of the formation of carbon dots made by citric acid and urea. This was done by using DFT calculation and molecular dynamic parametrization in order to have a clear idea of how each molecule which is formed during the synthesis can interact one with each other in order to give both the molecules or the carbon structure. So we are using some evaluation of the barrier required to get into those geometries. We need to consider some of the possible reaction that could happen during our synthesis, of course, and we are using a sort of molecular dynamics reactive in the presence of water, which was modeled by TIP 3P during the synthesis. So we have several pathways which are simultaneously present. And the network of the chemical reaction is quite complicated. And of course, we choose to focus only on few key reasonable starting reactions, like the one I'm showing you here. First, we started with the urea decomposition. Then we have the interaction of the citric acid with some residual part of the urea, like ammonia. And we have the first amination. We could have the first amination. After the first amination, we could have both second amination or cyclization. in this presentation, I will show you what happens after the first cyclization, but we are still studying what happens if the cyclization is realized after the second amination. And then after the cyclization, there could be the aromaticity of our system, so the aromatization of our system, which gives the citrazinic acid, in this case I'm showing you. And after the aromatization, there could be a second amination of the system, giving another molecules which is also emitting in the blue, like the citrazinic acid amine, or the formation of second ring, aromatic ring, to give the HPPT molecules, which is usually produced with a very high amount of urea. We are still working on that because we are working on system produced by different amount of citric acid and urea. So you see here our first results about what happens in the case of 1:1 molar ratio of citric acid and urea, and in the case of 1:10 citric acid and urea molar ratio. You see that in the first case we have the formation of citrazinic acid and its family at the expense of course, of citric acid and urea. And in the other case, there is also the formation of HPPT, which started after, as you see here, after the formation of citrazinic acid, which is here, which is what we indeed expected from the experimental point of view. And we asked ourselves, is this the case? So how can we monitor the formation of different molecules during the synthesis? We decided to perform the synthesis and stop the synthesis at different time. So we start from few seconds till 180 minutes, 3 hours, heating our samples at 180 Celsius degrees in order to get the formation of our molecules. And we look at them by using optical spectroscopy and NMR spectroscopy. So you see here that after the first minutes, we have the formation of some nice blue emitting molecules, and then at the end we have also the formation of something which is emitting the green. You see, this is blue because it's emitting at about 425 nanometers with an excitation of 350 nanometers, and this is green because it is emitting above 500 nanometers by exciting the sample above 400 nanometers, which means exciting the blue with the emission in the green. And if we look at the results of the NMR, which was performed in the solid state case in order to get information on the powders, and in the liquid state in order to get information about the molecules. So in one case we are looking preferentially to the structure of the carbon network. In the other one we are looking preferentially to the molecules which are on the surface of our system. We see that we have from the very first minutes, we have the formation of some aliphatic, some molecules in the aliphatic range, some signal in the carbonyl range. We have also the reduction of the citric acid as expected, and the formation of bonding between carbon and nitrogen, which suggests the formation of nitrogen species derived from the urea. So, by looking at these experiments, we are able to see that from the first minutes we have both the formation of molecules, but also the formation of the carbon nanostructure, the carbon network. I mean, we were able to separate, in collaboration with people from Czech Republic and Slovakia, we were able to separate our samples in order to get idea of the mass of the mass of our molecules and the NMR signal of this molecule for the identification, which is because it is important to understand from for our community if citrazinic acid is formed or if are formed other molecules which are citrazinic acid like. In this case, we have, for example, the amine in the amide form of the citrazinic acid and not the citrazinic acid by itself. And we were able to show this formation by the separation of our molecules in the systems. And we are also studying what happens if we increase the amount of urea. And you see in this case that starting from the blue, the systems at the end is completely green, since we have at the beginning the emission in the range of around 425, and at the end we have only emission in the range above 500 nanometers, which means that the blue molecules which are formed at the beginning, citrazinic acid-like molecules, are completely transformed in green emitting one like HPPT. We also performed the gel filtration chromatography in order to get the mass of HPPT and the liquid NMR signals of HPPT to have confirmation of the green emission that you see here in the separation. And also by solid state NMR and nitrogen 14 NMR, we are still working on that, but the information we get is more or less the same that we have before. So we performed time resolved DFT and in order to get an idea if these molecules could be responsible for the emission that we get from the experimental point of view, we considered both the neutral and the -1 species because we are in water, so we could expect in some case the formation of -1 species, of ionic species. We computed the optical absorption and the emission, so you see here that they could be blue or green depending on the species we are looking at, and they could explain the optical feature that we have seen from the experimental point of view. So the first conclusion it was that we can be able to tailor the optical properties of our carbon dot by changing the content of citric acid and urea. We can also achieve some predictiveness by using more accurate experimental characterization in order to give useful information for computational approach. And we need a proper model. When we have that proper model, we can solve advanced critical issues which are still open. For example, how do the fluorescent species bind to the graphitic core and why they are not affected by aggregation quench phenomena, because our emissions are quite efficient. And how was the pH of the solution important for the optical properties of carbon dot? Well, we started from the first critical issue we want to solve by a different approach, and we try to build our system. I will go a bit faster in this part because you will see a nice presentation from Zaira in this afternoon. You can have also some details there, but the idea is that we constructed our carbon nanodots by a builder. We perform the simulation in the ensemble NPT. The thermostat was at 300K, the barostat was at 1 atmosphere. We have water all around, and we put not only our carbon particle models, but also some of the molecules that we get from the experiment that I've shown you, in order to see how these molecules interact with our carbon nanostructure. So the idea is to look at the simulation, a molecular dynamic simulation, in order to have an idea if our molecules get trapped by the carbon nanostructure, and if this trapping is how it's made, I mean from the bonding of the molecules to the structure, and how long it lasts. So we constructed different carbon nanostructure with or without nitrogen. We put some molecules like citrazinic acid and citrazinic acid-like molecules in order to get the idea if they interact with our carbon dot. In the case of graphitic nitrogen or without nitrogen in the systems, we produced this kind of system, and we get the simulation lasting for about hundreds of nanoseconds in order to have to reach stability. We are still performing also some statistics, so changing the starting condition in order to get the idea if all the results are soundings. But what we have here is again the production of the carbon dots with the proper distance, at least in the case of graphite-like carbon nanodots. We have the presence, we observed that the presence of the fluorescence doesn't change the distance in between the layers, and that when the molecules are able to be trapped by our carbon nanodots, they are trapped on the surface and mimicking again the distance in between the layers, since these organic molecules are planar. And this was obtained both in the case of doped and undoped system. If we look at the radius of gyration, which gives us an idea of the stability of the system during the evolution, we get the radius of the gyration of the carbon dot pair, so without nitrogen, and the one of the with nitrogen, which are quite similar. What about when we put inside some molecules, where you see that in the case of the bare system, the radius of the gyration increases quite a lot, and the molecules stays on the carbon dots for a very short period. You see here from this part, which is flat but is higher than before, and also it's quite noisy. On the other hand, if we look at what happens in the case of a nitrogen doped carbon dot and the interaction with citrazinic acid-like molecules, what we get is that we have, yes, again, a radius of gyration larger than without molecules, but at the beginning, but we have the stabilization for a quite longer period of the system, where we observe the trapping of our molecules around the carbon nanodots. So the idea was also pursued by studying the hydrogen bonding in order to have an idea how these molecules are eventually related to by themselves or to the layers of our systems. Okay, is more or less the same in the case of citrazinic acid without molecules, or in the case of citrazinic acid with in the case of carbon dots with bare, or in the case of carbon dots with nitrogen. You see that also in the case of the carbon dots, we have in some cases we have the the trapping of the molecules, at least in the case of single molecules, because in the first case I've shown you there were different molecules altogether present at the same time. So these are just for statistics, and we are still doing that. The conclusion is that with an experimental approach, were able to study the optical properties, and with the modeling, we were able to understand how these molecules, which we are sure from the experimental point of view, they are formed. how this molecule can interact with a useful model of our carbon dot. In this case, we are considering only an ordered model. In the future, we want to consider also some disordered model of our carbon dots. But what we see is that the fluorophores are easily bonded through hydrogen bonding at the surface of nitrogen doped carbon dot, and they are more easily bonded in the case of nitrogen doping carbon in the carbon dot than in the case of carbon dots without nitrogen doping, which is again in agreement with experimental results where we have that the carbon dots doped with nitrogen are more efficient from the optical point of view. So we can say that we can design low-cost, green, and environmental-friendly luminescent materials by using a combined approach of experimental and computational chemistry. I want to thank all the people which were working in the group for this research, including people from Cagliari, Namur, Matej Bel, Slovakia, Olomouc in Czech Republic. In particular, I want to thank Francesca, Yael, Antonio, Zaira, and Chiara for the work they have done. I want to invite you to visit Cagliari in Sardinia. I'm showing you some nice picture of our university there. So thank you for your attention. I'm here for your question.
Thank you very much, Carlo, for this very nice talk. And okay, now it's the time for questions from the audience and from the Zoom. Perhaps Sergey.
Are there any questions in audience?
He's better than me.
Carlo Maria, thank you very much for your very interesting lecture. We have one question in Zoom from Andrei Baumkettner, I see.
All right, can I go ahead?
Yes,
yeah.
Please.
Oh, okay, thanks for very nice presentation. I was wondering if you have an idea what causes these shifts in your color in fluorophores. You show the graph in different media, DMSO, toluene, etc. Do you know what is the reason for the color change? Is that the polarity of the solvent, pH, or maybe something else, some specific interactions with the solvent molecules?
Yeah, this is a very interesting question. I'm not performing experiments right now in different solvents, but we know from the literature that these molecules can change the optical properties depending on the solvent we are using. We are mostly interested in water because of biological application, but changing the solvent is a interesting way to probe our system in order to have an idea if the molecule is outside, I mean on the external shelf of our core shell system, or if the molecule is inside. Because if it is inside, it was shown by Marketa Polianchova and the others that usually the solvent molecules cannot get inside because the distance in between the layers is too small. Even in the case of water, it's not possible. So in that case, if it is inside, only inside, we shouldn't have any interaction with the solvent. If it is outside, of course, we have the interaction with the solvent. But unfortunately, it's quite difficult to separate from the experimental point of view both the situation. So it's tricky. It's an interesting point. It's not so easy to solve it.
All right, thank you.
Thank you for the question.
Other questions?
I would like to ask Carlo Maria, can you tell something more about QMMM calculations that you have used? As I understood, it was something about 16 nanoseconds as a trajectory.
You mean, sorry?
Yeah, maybe above.
Above?
Yes.
Okay, this one, you
Yeah,
mean?
this one. That's right.
Because we perform different kind. Yeah, it was 16 nanoseconds, and we stopped when we get some plateau, as you see.
And the size of your system, you also had some water molecules, as I understand.
Yeah, if the size is about 5,000 molecules inside our system, in order to get the typical experimental concentration of citric acid and urea.
I wanted to
Lot of water and yes, please.
So you were showing some like fluorophores connected by hydrogen bonds.
Yeah.
But upon the excitation, then actually I mean, it's actually not well known and understood actually, but the charge in the molecule changes completely, and the hydrogen bond actually can be like dissociated.
Yeah, okay. I was showing here in this case that, or I don't remember, maybe here, that if you have, you see here, two molecules are on top of the carbon dots and they are interacting. If I look at the hydrogen bonding, I see that there is the bonding in between these molecules.
Yeah.
As you said, they are forming a sort of dimer. In this case, it's a heterogeneous dimer. And of course, the optical properties are different. So we need also to understand how the interaction in between molecules, same kind of molecules, different kind of molecules, can bring different optical properties to our system. I have to say that in this case, since it's a molecular simulation, we have not performed yet the calculation of the optical properties of these molecules, of these dimers and trimers that we could have. There are some examples we also have done in the past for citrazenic acid, and we have seen that by having the presence of different dimers, we have a redshift of the optical properties. So they are a good option for our system in order to move the emission from the blue to the green, then to the red, which is what we want to have for biological application. But we still have to understand how they are, how it is working really. So,
Okay. Sonia, you had a question?
Yes. Good afternoon. So thank you, Carlo, for a very nice presentation. I have a very small question. It's about force field modeling. You mentioned that you use amber force field, and I'm just curious, how did you obtain the charges for this modeling?
Well, I'm not done by myself, so I don't know the detail, but I'm sure if you ask this question to Zaira in the afternoon, she can give you the information.
Or maybe some rest fitting.
Yeah, I suppose so, but I don't know the detail, really. Sorry for that.
Okay, thank you.
Okay, other questions? I cannot see more questions. But already we had several.
There is a question in the chat. The question is, what is the role of nitrogen doping? Can nitrogen doping helps to produce red dot? Why red dots are hard to get?
Okay, nitrogen doping is done on carbon dots because we know from the experimental point of view that we get usually more efficient carbon dot in the terms of the emission. The presence of nitrogen, if engineered, can shift till the green, usually the emission properties of your molecules. To go to the red, we need some different dopants. But the idea is that using dopants allows you to produce different molecules which could emit in the green, yellow, or in the red. The problem with the red is that, well, from the experimental point of view, Carbon dots made by precursors and having red emission, usually they have a very poor quantum yield. So they are not so useful at the moment for biological application because you need a lot of light in order to get imaging. We are solving the problem by starting with molecules already formed that we know emit in the red. For example, safranin. And in that case, the molecules are attached, bonded to the surface. The optical properties are not changing so much with respect to the isolated molecule in solvent. So it was an approach useful, but from my point of view, is different. I mean, I'm not starting from the precursor to get something which is red. I'm starting from a precursor which is red by itself. But the idea to have red one is that they could be very, very useful for bioimaging application. Why we are going to a bioimaging application? Because we want to avoid the use of quantum dots. Quantum dots are toxic because of the presence of cadmium, for example, or lead. And we do not want to have cadmium and lead in our blood. So we need to substitute these probes by different ones, and carbon dots could be a very not toxic and green solution. I hope I've answered to your question.
Piotr?
Okay. Yes, so I think there are no more questions according to what I see. So let's thank Carlo again and for this nice contribution, and once more let's thank both speakers for a nice session. And
Piotr, thank you very much.